So imagine a surgical oncologist just walks out of the operating room. They've just completed this really grueling resection for a patient with hepatocellular carcinoma.
Right, liver cancer.
Exactly. And the margins are clear, the scans look pristine. I mean, by all standard metrics, the tumor is gone and the patient is functionally cured.
Functionally, yes, on paper.
Right, on paper. But underneath that clinical victory is this, statistical terror. Within 5 years, up to 70% of these patients will experience a recurrence.
Yeah, it's a huge number.
And for the vast majority of them, that recurrence happens violently within the first 2 years. So we're looking at a cancer that doesn't just return, right? It comes back with an absolute vengeance. Welcome to the Deep Dive, by the way. I'm your host, and as always, I'm joined by our resident expert to unpack all of this.
Glad to be here. And, that 2-year window you mentioned, that is the clinical nightmare we call early recurrence.
Early recurrence, okay.
Yeah, and the driving force behind it is often something called microvascular invasion, or MVI. So the surgeon removes the primary mass, but these microscopic clusters of malignant cells have already hijacked the tiny blood vessels surrounding the liver.
So they're completely invisible.
Entirely invisible. You can't see them with the naked eye, and traditional imaging misses them completely.
It really reminds me of tending a garden. You can dig deep, grab a really stubborn weed by the stem, and pull out the entire visible plant. The soil looks perfectly clear.
But the roots are still there.
Exactly. If you leave even a fraction of a highly energized root system behind, it's going to tap right back into the soil's resources. And within weeks, the weed hasn't just returned, it has completely colonized the space.
That's a perfect analogy.
So today, for you listening, you are going to want to take notes, because our mission is to look at the exact molecular engine powering those hidden roots. We are unpacking a fascinating 2026 paper from Cell Death and Disease that maps out, protein by protein, how these residual cancer cells rewire their own power grids to survive.
And the researchers approached this with absolute rigor. I mean, they didn't just look at a few cell lines in a dish. They started with a retrospective clinical cohort of 140 patients.
Wow, 140.
Yeah, they were hunting for a specific biomarker that could predict this early recurrence. And they actually found an independent predictor of both poor disease-free survival and overall survival.
But to find that predictor, they had to do this incredibly sophisticated molecular suspect lineup, right? Because like you said, the culprit is microscopic. You can't just look at the tissue.
Right. You have to look at the protein expression driving the tissue. So they use something called label-free quantitative proteomic profiling.
Okay, walk us through that. How do they actually narrow down the suspects?
Well, they took tumor tissues from 6 patients who experienced early recurrence, which they call ER, and 6 who had no recurrence, or NR. And they matched those with healthy adjacent liver tissue.
Got it.
Then they ran these samples through a mass spectrometer, which basically lets them identify the exact proteins present based on their molecular mass. So the initial scan yielded an astonishing 6,856 proteins.
Which is essentially mapping the entire active biochemical universe of those cells. That's massive.
It really is. So to find the specific engine driving the aggressive return, they had to filter for what was fundamentally out of place.
Right.
They isolated 1,209 differentially expressed proteins between the tumors and the healthy tissue.
Okay, narrowing it down.
Then they cross-referenced that list, looking specifically for proteins that were heavily upregulated, meaning, the cell was mass-producing them in the early recurrence tumors compared to the non-recurrence ones.
And that gets them to the final list.
Yeah, that filtered the list out of 49 key proteins.
And this is where the paper throws a massive curveball, because when they mapped the subcellular localization of those 49 proteins, like where they actually live and work inside the cell, all the data pointed straight to the mitochondria.
Specifically to a protein called MRPL3.
Okay, wait, I have to push back on this entirely.
I suspect I know where you're going with this.
Well, yeah, because we've done deep dives on cancer metabolism before, and the cardinal rule of tumor growth is the Warburg effect. We are taught that cancer cells are fundamentally greedy for sugar.
Right, they love glucose.
Exactly. They bypass the mitochondria almost entirely, and they rely on a highly inefficient but incredibly fast process called glycolysis to just ferment sugar and grow rapidly. So if tumors prefer to shut down their mitochondria, why are these ultra-aggressive, early recurring cells suddenly relying on the cell's traditional power plant?
It's a brilliant contradiction, really. And it highlights the sheer metabolic plasticity of cancer. You see, the Warburg effect is a hallmark of many primary tumors, sure, but it is not a universal law.
So they adapt.
Exactly. Think about the microenvironment of a liver immediately post-surgery. It is hypoxic, inflamed, and severely nutrient-deprived.
Oh, right. The blood supply is messed up.
Exactly. So rapid, inefficient glycolysis isn't going to cut it when resources are that scarce. These microscopic residual cells don't abandon the mitochondria, they supercharge them.
Wow.
They rely on oxidative phosphorylation, or ALK-XPHOS, to extract every possible molecule of ATP from their environment.
Okay, so I'm tracking the biology here. The clinical reality is that the surgery leaves these microscopic cells behind. And instead of starving in the damaged tissue, they switch their metabolic strategy.
They pivot.
Right. They lean into hyperactive mitochondria to survive. And the primary red flag for this activity is elevated MRPL3.
That's the one.
It makes you wonder how often in medicine, or, any complex system really, we assume a problem is caused by a known mechanism, only to realize the system has completely flipped its strategy to survive our interventions.
It's a scary thought. And before we get into exactly how MRPL3 does this, let's just do a quick recap. We're at the 5-minute mark here.
Good idea. So bullet point 1, HCC often returns early due to hidden microscopic tumor cells in the blood vessels.
Bullet point 2. The researchers used proteomics and identified MRPL3 as the primary suspect linked to this aggressive return.
And bullet point 3. Instead of the expected warbird effect, where they just ferment sugar, these specific tumors rely on supercharged mitochondria to survive.
Exactly.
So for you listening, think about this. Think about a time you assumed you knew the root cause of a problem, only to find the actual mechanism was doing the exact opposite of what you expected. How did you pivot?
That's a great question to keep in mind, because that metabolic pivot is exactly what makes early recurrence so lethal. And to understand how the cancer pulls this off, we have to look closely at what MRPL3 actually does.
Right. Let's get into the mechanics.
So MRPL3 stands for mitochondrial ribosomal protein L3. It is a structural anchor of the 39S large ribosomal subunit inside the mitochondria.
Okay, let's visualize that. If the mitochondrion is a power plant, the mitoribosome is the assembly line building the machinery for the plant, right?
Yes.
So MRPL3 acts as this indestructible factory foreman on that assembly line.
I like that. A foreman who is responsible for translating 13 vital mitochondria-encoded proteins. These include things like ND1 and COXI.
And what do those do?
They are the physical complexes in the electron transport chain. They are the molecular turbines that pump protons and generate cellular energy. And the researchers brought an exhaustive battery of tests to prove just how much control this foreman has over the cancer's survival.
Yeah, they didn't just observe the cells, they actively sabotaged the assembly line to see what would happen.
Right, in vitro testing.
Exactly. When they used genetic tools to silence the MRPL3 gene in human HCC cell lines, like Malavu and HIH7 cells, the results were catastrophic for the tumor.
The cancer cells literally lost their ability to multiply.
Yeah, in the CCK8 assays, their proliferation just tanked. And when the researchers scratched a line through a culture of these cells for a wound healing assay, the cells couldn't migrate to close the gap.
Their invasive properties completely shut down because their energy grid was severed. You can see in the Transwell assays too.
But conversely, when they forced the cells to overexpress MRPL3, the cancer just went into hyperdrive.
Oh, absolutely. They tested this in vivo in subcutaneous xenograft mouse models, basically injecting human HCC cells directly under the skin of the mice.
And the results.
The tumors with elevated MRPL3 grew into massive volumes, and the tissue stained intensely for Ki-67.
Which is a classic biological marker that indicates rapid, uncontrolled cell division, right?
Exactly. So we know the foreman is driving the growth, but the researchers wanted to know the exact mechanics of how the factory was operating under this foreman's direction.
So they looked at the actual energy output.
Right. They used high-resolution respirometry, specifically a machine called the Ouroboros O2K.
Ouroboros O2K sounds like a sci-fi ship.
It does. But it actually measures the exact oxygen consumption of the cells in real time. And the data is stunning. MRPL3 doesn't just bump up energy production, it overhauls the entire capacity of the system.
Oh, so?
It significantly increases basal respiration, which is the engine's idling speed. It increases maximal respiration, pushing the engine to its absolute limit. It boosts ATP-linked respiration, meaning the actual output of usable energy spikes.
Wow.
And most importantly, for a cancer cell under attack, it drastically increases the spare respiratory capacity.
Okay, so it gives the cell an emergency reserve to handle sudden metabolic stress, like chemotherapy or hypoxia.
Exactly.
But here is the paradox. In cellular biology, running the mitochondrial engine at absolute maximum capacity usually creates a lethal amount of exhaust. We're talking about reactive oxygen species, or ROS.
Yes, ROS is highly toxic. It's the unavoidable chemical exhaust of the electron transport chain.
Right, it's just a byproduct of making energy.
And when ROS levels get too high, they oxidize lipids, they shred mitochondrial DNA, and they trigger a cascade that ends in apoptosis. The cell essentially self-destructs to prevent the damage from spreading.
So by all logic, these hyperactive cancer cells should be choking on their own pollution.
They aren't. And this is where the genius of MRPL3 comes in. The researchers used fluorescent dyes like JC1 that shift color based on mitochondrial health.
To measure the membrane potential.
Right. And they found via FACS analysis that MRPL3 dramatically increases the mitochondrial membrane potential. But when they measured the cellular and mitochondrial ROS levels, they were inexplicably low.
That is wild.
MRPL3 isn't just telling the factory to produce more energy, it is actively upgrading the exhaust vents so the cell is protected from its own toxic byproducts.
And we can actually see this structural upgrade. Right.
Yes. They use transmission electron microscopy, or TEM, to look at the cristae.
Those are the complex inner folds of the mitochondria where the electron transport chain physically sits.
Exactly. In cells without MRPL3, those folds are swollen, disorganized, and fragmented. The infrastructure is literally collapsing. But in the cells overexpressing MRPL3, the cristae are robust, dense, and tightly packed. The factory floor is immaculate and highly efficient.
So MRPL3 turbocharges cell energy production while protecting the cell from toxic ROS damage, which directly fuels cancer spread. Let's do a quick 10-minute recap.
Sounds good. Bullet 1. MRPL3 is the mitochondrial hormone supercharging ATP and lowering toxic ROS.
Bullet 2. We know it creates this incredibly efficient factory, but we still need to figure out how it communicates these upgrades to the rest of the cell.
And for you listening, here's a question. If you could upgrade one, exhaust vent in your own daily workflow to prevent burnout, what would it be?
Oh, I love that. Think about your own structural upgrades. All right, let's tie these threads together. We have this lethal, early recurring liver cancer. It survives surgery by hiding in the microvessels, and it survives the harsh environment by turbocharging its mitochondria.
With MRPL3 as the foreman.
Right, maximizing usable energy while somehow scrubbing the toxic exhaust. But if you think about the architecture of a cell, the amount of coordinated communication required to pull off that dual function is staggering, which led the researchers to their next major hurdle. How is MRPL3 actually calling in these upgrades?
Yeah, they needed to find the biochemical algorithm at play. What is MRPL3 interacting with to trigger this massive structural shift?
So they went hunting for a secret handshake.
Exactly. They used a technique called immunoprecipitation coupled with liquid chromatography, tandem mass spectrometry, or IPLC, MSMS for short.
That is a mouthful. Basically, conceptually, it's like using molecular tweezers designed to grab only MRPL3, right?
Yes. They pull it out of the cellular mixture and analyze whatever other proteins are physically stuck to it. And the protein shaking hands with their foreman was TOM-M40.
TOM-M40. Okay, what is that?
It's a core structural subunit of the translocase of the outer membrane. It acts as the primary gatekeeper on the exterior wall of the mitochondria, responsible for recognizing and importing essential nuclear-encoded proteins into the mitochondria.
And the mapping they did to prove this interaction is incredible. They didn't just observe the two proteins near each other, they systematically dismantled them to find the exact interlocking puzzle pieces.
Right, truncation mapping.
Yeah. They sheared off different segments of the amino acid chains, and they proved that a very specific structural domain, the delta R1 region, which spans amino acids 1 to 174 of MRPL3, binds directly and exclusively to the delta R2 region of TOM-M40, which sits between amino acids 181 and 361.
It's like finding the exact teeth on the key that open the lock. Mm-hmm. And the precision of that mapping is crucial because it tells us how the foreman is manipulating the gatekeeper.
Right.
MRPL3 isn't signaling the cell's nucleus to print more TOM-M40 mRNA. It's not manufacturing new gates. The binding itself acts as a physical shield for the gates that already exist.
Okay, so it stabilizes TOM-M40, and the methodology here is fascinating. They hit the cells with cyclohexamide,
which is a drug that completely halts all new protein synthesis. The factory's frozen.
Right. Under normal conditions, the existing TOM-M40 proteins would just quickly degrade. But with high MRPL3 levels, TOM-M40 just stuck around. Its half-life was significantly prolonged.
And they further confirmed this by introducing a proteasome inhibitor, MG132. The proteasome is basically the cell's garbage disposal system.
It shreds old or damaged proteins?
Exactly. When they shut down the garbage disposal, TOM-M40 levels remain high regardless of MRPL3.
So that proves MRPL3's primary role in this handshake is to physically block the cell from degrading its mitochondrial gatekeepers.
Yep, it acts as a shield.
Okay, but I have a major spatial pushback here, a biological paradox.
Let's hear it.
We established that TOM-M40 is the gatekeeper embedded in the outer membrane of the mitochondria, right?
Correct.
But MRPL3 is part of the mitoid bosom, which lives deep inside the mitochondrial matrix, past the inner membrane. There is physical space, the inner membrane space, separating them. How in the world are they shaking hands? Are these cancer mitochondria literally squished together?
It's a brilliant observation, and it touches on one of the most cutting-edge concepts in the paper. The researchers hypothesize that this interaction relies on contact site microdomains.
What does that mean?
Well, in a healthy, unstressed cell, the inner boundary membrane and the outer membrane maintain a distinct architecture, their space. But cancer fundamentally bends the physical rules of the cell to survive.
So the intense metabolic stress of the tumor microenvironment forces the mitochondria to warp their own geometry.
Exactly. The membranes pinch together, creating these disorganized contact sites where the matrix-dwelling MRPL3 is suddenly brought into direct physical proximity with the outer membrane TOM-M40.
Wow.
The cancer forces a structural anomaly to facilitate a survival handshake that shouldn't normally happen.
That is staggering. The tumor is warping the internal architecture of its organelles just to establish this chain of command.
It's doing whatever it takes.
So to recap our biochemical pathway up to this point, MRPL3 acts as the mitochondrial foreman, reaching across these warped membrane contact sites to physically stabilize TOM-M40, the outer gatekeeper.
Right.
This ensures the mitochondria can continue to import whatever resources they need without the gatekeepers degrading. But how does that stabilization translate into the massive energy spike in tumor growth we discussed earlier?
Well, this brings us to the final axis of the mechanism. The researchers ran RNA sequencing on the normal cells versus the MRPL3 overexpressing cells, looking at the entire transcript.
Now, what did they find?
They found a massive enrichment in AMPK signaling and specifically mitochondrial biogenesis pathways. The stabilization of TOM-M40 triggers a major downstream surge in a protein called PGC1 alpha.
And PGC1 alpha is essentially the master switch for cellular metabolism and mitochondrial biogenesis, right? When it gets flipped, the cell knows it's time to build more energy infrastructure.
Precisely. And PGC1 alpha, in turn, aggressively upregulates TFAM, which stands for mitochondrial transcription factor A.
Okay, TF.
TFAM is the mechanic that actually executes the order. Its job is to drive the replication, transcription, and maintenance of mitochondrial DNA.
So the loop completely closes. You have MRPL3 stabilizing TOM-M40. TOM-M40 spikes PGC1 alpha. PGC1 alpha activates TFM, and TFM physically builds out more mitochondrial capacity,
giving the cancer cell limitless clean energy to spread through the microvasculature.
It is a highly efficient, tightly controlled biological assembly line, but, because it relies on such a strict chain of command, it operates exactly like a Jenga tower.
That's the perfect way to look at it. And that rigid structure is its ultimate vulnerability.
So if you pull out one block, does the cancer's advantage collapse?
They systematically tested this by pulling out individual blocks to see if the tower would hold. Using small interfering RNA, they knocked down either TFAM or TOM-M40, these supercharged aggressive cancer cells. The moment one of those downstream links was broken, MRPL3 superpowers were completely neutralized.
The massive oxygen consumption we talked about earlier just plummeted right back to baseline, and the toxic ROS exhaust, which MRPL3 had so carefully scrubbed, suddenly spiked because the internal exhaust vents were broken.
The Jenga tower crashed, taking the cancer cell's energy grid down with it. The invasive properties ceased entirely.
But the researchers didn't stop at petri dishes, did they?
No, they brought this directly into clinical translation. They used an orthotopic liver tumor model in mice.
So instead of just injecting cancer cells under the skin like before, they surgically implanted the MRPL3 overexpressing cells directly into the mouse livers.
Yes, perfectly mimicking the actual anatomical and metabolic environment of hepatocellular carcinoma.
A far more rigorous test of the tumor's ability to survive in its native habitat.
And once the aggressive tumors were established, they treated the mice with oligomycin,
which is a potent OXPHS inhibitor. It physically binds to and blocks the ATP synthase machinery in the mitochondria.
By administering this drug, they essentially pulled the foundational block from the Jenga tower. The massive tumor volume and growth driven by MRPL3 was completely suppressed.
They starved the cancer of its specialized power source.
Exactly. So, time for our final recap.
Right. So, bullet point 1. The MRPL3 advantage relies on a strict chain of command, the TOM-M40 to PGC1 alpha to TFAM axis.
Bullet point 2. Breaking any link in this chain starves the cancer.
Which leads to a question for you listening. Where in your life or work do you have a Jenga tower process? You know, a chain of events so dependent on one another that removing a single step causes the whole thing to fail.
That's a good one to reflect on.
So let's bring all of this exhaustive molecular mapping back to the patient sitting in the oncology clinic. For decades, we've treated cancer metabolism as a monolith, assuming they all just ferment sugar.
But we now have concrete proof that some of the most dangerous early recurring tumors are actually sophisticated mitochondrial powerhouses.
And the implications for personalized oncology are profound. Currently, navigating post-surgical treatment for HCC can feel like, well, a statistical waiting game.
But imagine if, immediately following a resection, oncologists routinely profiled the excised tumor tissue. If they screen the proteomics and find elevated levels of MRPL3, they immediately know two crucial things.
Right. First, this patient is at extremely high risk for a violent 2-year early recurrence via microvascular invasion.
And second, they know exactly how to target the residual cells. You don't just blast the patient with standard broad-spectrum chemotherapy that targets rapidly dividing cells.
You target the power grid, you utilize OXPHOS inhibitors to pull the plug on the mitochondria, collapsing that specific metabolic advantage before the microscopic roots can take hold in the liver.
It is the definition of precision warfare at the molecular level. It transforms our understanding of recurrence from a random, unlucky event into a predictable, mechanical process that we can interrupt.
It fundamentally changes the game. But as we wrap up this deep dive, I want to leave you with a mind-bending final thought that stems directly from this research. The entire lethal advantage of this cancer relies on a spatial anomaly.
That contact site microdomain.
Exactly. It relies on the internal matrix-dwelling MRPL3, reaching across a warped, disorganized contact site to shake hands with the outer membrane TOM-M40.
A physical bridge built purely out of the stress of the tumor microenvironment.
So, what if the future of advanced cancer therapy isn't about designing toxic chemicals to destroy these proteins at all? What if, instead, pharmacology moves toward architectural medicine?
Architectural medicine. I love that phrasing.
If we can develop compounds that simply force the inner and outer mitochondrial membranes to maintain their normal cellular geometry, that handshake becomes physically impossible.
We wouldn't have to fire a single chemical shot at the cancer's internal machinery. By simply restoring the normal physical space between the membranes, the chain of command is broken.
The gatekeeper degrades, PGC1 alpha never spikes, and the power plant shuts down. You defeat the cancer just by fixing the shape of the room.
It brings us right back to your garden analogy.
Yeah, exactly. If you want to stop that aggressive, hyper-energized weed from coming back, maybe you don't need to invent a stronger, more toxic poison to pour on the roots. Maybe you just need to subtly change the structural composition of the soil so the roots can no longer communicate with each other.
It's an incredible shift in how we view the physical battlefield of disease.
Definitely something to mull over as you go about your day. Thanks for joining us on this deep dive.